Hello everyone, this is Professor Beda Ahmed, uh, and we are in chapter 5. I will cover the structure and function of large biological molecules. Uh, so, we are going to be talking about biomolecule, or otherwise known as macromolecules. So these are the things that make up living things. So the crux of macromolecules comes down to carbon atoms. So these are all organic molecules, which means that it contains carbon. Now remember that carbons can is super versatile. It can bond with itself, it can bond with hydrogen, oxygen, nitrogen, anything, and form complex, large structure. Uh, the molecules of life, all living things are made up of four classes of large biological molecules: carbohydrates, lipids, protein, nucleic acid. And I will cover carbohydrates and lipids in this lecture. Uh, so, macromolecules are large molecules and are complex. Large molecules have unique properties that arise from orderly arrangement of their atoms. Carbohydrates uh, are a source of energy and provide structural support. Proteins have a wide range of functions and transporting substances into and out of the cell. Nucleic acid uh, store genetic information and function in gene expression. Lipids are a group of diverse molecules that don't mix well with water. The key function includes providing energy, making cell membrane, and acting as hormones. A polymer is a long molecule consisting of many similar or identical building blocks linked by covalent bonds, much as train consists of a chain of boxcars. The repeating units that serve as the building blocks of a polymer are smaller molecules called monomers. Okay, this is from Greek words. Mono means single, poly mean many, and mers mean units or parts. Although each class of polymer is made up of different type of monomers, the chemical mechanism by which cells make polymerization, and break them down are similar for all classes of large biological molecules. In cells, these processes are facilitated by enzymes. Enzymes are specialized macromolecules, usually proteins, that speed up chemical reaction. A reaction that connects a monomer to another monomer, or a polymer, is a condensation reaction. This is in general. Okay. If a reaction in which two molecules are covalently bonded to each other with the loss of a small molecule, if water molecule is lost, it is known as a dehydration reaction. So in this reaction, one molecule of water will be lost or released from this reaction to form the bond between monomers. For example, carbohydrates and proteins polymers are synthesized by dehydration reaction. Each reactant contributes parts of the water molecule that is released during the reaction. One provide a hydroxyl ion, while the other provide hydrogen ion. This reaction is repeated as monomers are added to the chain one by one, lengthening the polymer. Polymers are uh, disassembled to monomers by hydrolysis, this reaction. This a process that is essentially the reverse of the dehydration reaction. Hydro mean water, lysis mean breaking. Okay, so hydrolysis means water breakage. The bond between monomers is broken by the addition of water molecules. In this process, with the hydrogen ion uh, from one water attaching to the one monomer and hydroxyl ion attaching to the other. We have an example of hydrolysis reaction within our bodies is the process of digestion. The bulk of organic material in our food is in the form of polymers that are much too large to enter our cells. Within the digestive tract, various enzymes attack the polymers, speeding up hydrolysis, release monomers, and are then absorbed into the bloodstream for distribution to all body cells. Those cells can then use a dehydration reaction to assemble the monomers into new different polymers that can perform specific functions required by the cells. Okay. Carbohydrates include sugars and polymers of sugars. The simplest carbohydrates are monosaccharides. Those are the monomers or simple sugars. These are monomers from which more complex carbohydrates are built, meaning that disaccharides and polysaccharides made up from monosaccharides, okay? They made the other type of carbohydrates, the more complex carbohydrates. Disaccharides are double sugars consisting of two monosaccharides joined by equivalent bond. Carbohydrates, macromolecules are polymers called polysaccharides composed of many sugar building blocks. Uh, Glucose is of central importance in the chemistry of life. In the structure of glucose, we can see the trademarks of monosaccharides. Um, the molecule has a carbonyl and many hydroxyl ions in there. So these two most important are the trademarks for any sugar. Any sugar contain two functional group, carbonyl group, and many hydroxyl group. Okay, uh, we can classify or depending on the location of carbonyl, we can classify the monosaccharides or naming them. You remember that if the carbonyl group is uh, is at the end of glucose or the sugar, we call this aldose. If the carbonyl group within the carbon skeleton, it's ketone. Okay, so glucose is aldose. Why? Because the carbonyl group at the end of the carbon skeleton. Fructose, isomers of um, glucose, uh, the carbonyl group is within the carbon skeleton, so fructose is aldose, ketone. So glucose is aldose, fructose is ketone. Another criterion for classifying monosaccharides is the size of carbon skeleton, which ranges from 3 to 7 carbons long. So glucose, fructose, and other sugar that have 6 carbons are called hexose. If they have 3 carbons, those are triose. If they contain 4 carbon tetrose, um, 5 pentose, 6 hexose, 7 heptose. Another source of sugar diversity for simple sugar, of course, is in the way their parts are arranged, especially around symmetric carbons. Recall that a symmetric carbon is a carbon attached to 4 different atoms or a groups of atoms. Glucose, galactose, as we see here, glucose and galactose, for example, differ only in the placement of parts around one symmetric carbon. Uh, See the purple boxes here. What seemed like a small difference is significant enough to give the two sugars distinctive shapes and binding activities, thus different behaviors. Although it's convenient to draw glucose with a linear carbon skeleton, this representation is not completely accurate. In aqueous solution, glucose molecules, as well as most other 5 and 6 carbon sugars, form rings because they are the most stable form under physiological conditions. Monosaccharides, particularly glucose are uh, major nutrients for cells. In the process known as cellular respiration, cells extract energy from glucose molecules by breaking them down in a series of reactions. Not only are monosaccharides a major fuel for cellular work, but their carbon skeleton also serve as a raw material for the synthesis of other types of small organic molecules, such as monosaccharides, amino acids, and fatty acids. Disaccharides consist of two monosaccharides joined by glycosidic linkage. This is the name of equivalent bond between the monomers uh, of, uh, of the polymers of carbohydrates. So the type of covalent bond in carbohydrates is glycosidic linkage. So this is a covalent bond formed between two monosaccharides by a dehydration reaction. Glycomine sugar or carbohydrate or refers to the carbohydrates. And we have an example, maltose is a disaccharide formed by linkage of two molecules of glucose. In this example, we have sucralose. This is table sugar. It is two monomers are glucose and fructose. We have lactose, the sugar present in the milk, lactose. It's composed of glucose and galactose. Maltose, two glucose, two monosaccharides. So disaccharides is a molecule or macromolecule composed of two monomers or two monosaccharides. Okay. Polysaccharides are macromolecules, polymers with a few hundreds to few thousands of monosaccharides joined by glycosidic linkage. Some polysaccharides serve as storage material, hydrolyzed as needed to provide monosaccharides for cells. Other polysaccharides serve as building material for form structures that protect the cell or the whole organism. And then the architecture and function of polysaccharides are determined by its sugar monomers and the position of its glycosidic linkage. Both plants and animals store sugars for later use in the form of storage polysaccharides. Okay, so storage polysaccharides, there are stored carbohydrate for energy, okay, for later use. Plants store starch, a polymer of glucose. So each green pentose sugar is glucose. Okay, those are glucose, represent the monomer of this polymer. The polymer here is starch. Starch is a carbohydrate. So this is the polymer, consists of glucose monomers. So the plant stores starch. This is a polymer of glucose, okay, monomer, as granules within a cellular structure known as plastids. Plastids in potato tubers and grains, maize or corn, rice, those are the major source of starch. The simplest form of starch, amylose. Okay, amylose is unbranched, and the more complicated starch is amylopectin. Amylopectin is branched. You see the amylopectin here is branched. Both of them is a starch, but the starch divided into two types. One of them is unbranched amylose, and the branched one is amylopectin. Okay. Uh, Animals store a polysaccharide uh, called uh, glycogen. Okay, glycogen stored. A polymer is, glycogen is a polymer of glucose that is like amylopectin, but more extensively branched. Okay, is more branched, more than amylopectin. Okay, uh, vertebrates store glycogen mainly in liver and muscle cells. Breakdown of glycogen in these cells releases glucose when the demand for energy increases. Okay, and that's why storage polysaccharides, those stored for later use, those for using inside the cells or our bodies. Organisms build strong materials from structural polysaccharides. For example, the polysaccharides called cellulose. Cellulose, cellulose is a major component of the tough walls that enclose the plant cells. Okay, the enclosed plant cells protect the plant cells. Like starch, cellulose is polymer of glucose too, but the glycosidic linkage differ, which give the two molecules distinct three-dimensional shapes. So starch molecules are largely helical, as you see here. The starch molecule is helical. Cellulose never branched. Okay. Let's see here in plant cell wall. This is a plant cell wall. Uh, the parallel cellulose molecule here together in this way. Okay. They arranged into units called microfibril. Microfibril. These cable-like microfibers are a strong building material for plants and an important substances for humans because cellulose is the major constituent of paper and the only component of cotton. In fact, few organisms possess enzymes that can digest cellulose. Almost all animals, including human, don't. The cellulose in our food passes through the digestive tract as insoluble fiber. fibers. Along the way, the cellulose breaks the wall of digestive tract and stimulate the lining to secrete mucus, which aids in the smooth passage of food through the tract. A cow harbors cellulose digesting prokaryotes and protists in its gut. These microbes hydrolyze the cellulose of hay and grass. Uh, termite. Termite, which is unable to digest cellulose by itself, has prokaryotes or protists living in its gut that can make a meal of wood. Another important structural polysaccharides is chitin. Chitin, the carbohydrate used by arthropods, insects, spiders, crustaceans, and related animals to build their exoskeleton. Okay, exoskeleton, those are hard case that surround the soft part of an animal. Chitin is also found in fungi, uh, which use this polysaccharide as a building material for their cell wall. Lipids are the one class of large biological molecules that doesn't include true polymers. Okay, and they are generally not big enough to be considered macromolecules. The compounds called lipids are grouped with each other because they share one important trait. They are hydrophobic. They mix poorly, if at all, with water. Lipids consist mostly of hydrocarbon region with relatively nonpolar carbon-hydrogen bonds. They include waxes and certain pigments, but we will focus on the types of lipids that are most important biologically. We have the most important fats, phospholipids, and steroids. Orthofats are not polymers. They are large molecules assembled from molecules by a dehydration reaction too. Remember the dehydration, D mean remove, by hydration mean water, by removing water. So as we see here, uh, fat is composed of glycerol and fatty acids. Okay, glycerol is 3-carbon alcohol with hydroxyl groups attached to each carbon. Fatty acid uh, consists of a carboxyl group attached to, this is a carboxyl group attached to a long carbon skeleton. Uh, the molecules, the molecule building block of a fat are one molecule, glycerol, and three molecules of fatty acids. The carbon of the fatty acid are arranged in zigzag. Okay. Uh, in making fat, each fatty acid molecule is joined to glycerol by dehydration reaction. This results in ester linkage. We call the link or the type of covalent bond between fatty acid and glycerol ester linkage. So ester linkage found in, or the type of covalent bond in lipids are ester linkage. In carbohydrate, we call it glycosidic linkage. In fat, ester linkage. Okay. So ester linkage, a bond between hydroxyl group and carboxyl group. Okay, between the glycerol and fatty acid. Why it's triglyceride? Triglyceride because one glycerol will bond with three hydrocarbons by forming three ester linkage. We call this type of reaction dehydration reaction to form the bond between fatty acids and glycerol. Okay. Fatty acids vary in length, number of carbons, and in the number and locations of double bonds. The term saturated and unsaturated fat are commonly used in the context of nutrition. The term refers to the structure of the hydrocarbon chains of the fatty acid. Okay, we will focus on the hydrocarbon chain of the fatty acid. If there are no double bonds between carbon atoms composing a chain, then as many as hydrogen atoms as possible are bonded to the carbon skeleton. Such a structure are said to be saturated with hydrogen, and the resulting fatty acid therefore called saturated fatty acids. So this is a saturated fatty acids. Since there are no double bond between the carbon that form the carbon skeleton of the fatty acids. This type of fatty acids is saturated fatty acid. It's saturated with hydrogen bonds. Okay, each symmetric carbon bonded with two hydrogen. Okay, so this type of fatty acid is saturated fatty acids. Okay, this is a saturated fatty acid. At room temperature, the molecules of saturated fatty acids, or for the fat, saturated fat, such as the fat in butter, are packed closely. They packed closely. The tails they packed closely together, forming a solid. Okay, and that's why a butter is solid. Since there are no double bonds between the carbon that form the hydrocarbon of fatty acids. So saturated and unsaturated refers to the structure of the hydrocarbon chains of the fatty acids. So we describe the fat if it's saturated or not, depending on the if there's double bond or not between the carbon that form the hydrocarbon chains of fatty acids. An unsaturated fatty acid has one or more double bonds uh, with um, with one fewer hydrogen atom on each double bonded carbon. So unsaturated fatty acid has one or more double bonds with one fewer hydrogen atom on each double bonded carbon. Okay, at room temperature, the molecules of unsaturated fat, such as olive oil, cannot pack together closely enough to solidify because of the kink. We call this the kink because of the kink in their fatty acid hydrocarbon chain, and that's why uh, they can't pack closely together. Those the fatty acids. Nearly every double bond in naturally occurring fatty acid is a cis double bond, which creates a kink in the hydrocarbon chain. We call this cis, okay? Because the two hydrogen atom that bonded to the two carbons that form the double bond between them, they found on the same side on the double bond. We call this uh, cis, cis double bond. A diet rich in saturated fatty acids or fats may contribute to cardiovascular diseases known as atherosclerosis. Deposits called plaques develop within the walls of blood vessels. We have this process when they manufacture um, processed oil. This process called hydrogenation. In this process of converting unsaturated fats to saturated fats by adding hydrogen. Okay, hydrogenating vegetable oils also creates unsaturated fats with trans double bonds. This type of bonds, uh, each hydrogen atom found on different sides around the two carbon bonded, double bonded. Uh, so these trans fats may contribute more than saturated fats to cardiovascular disease. In addition to storage energy, adipose tissue also cushions such vital organs as the kidneys, and a layer of fat beneath the skin insulate the body. This subcutaneous layer is especially thick in whales, seals, and most other marine mammals, insulating their bodies in cold ocean water. Cells couldn't exist without phospholipids. So this is the second type of fats, lipids. We discussed the fat and now phospholipids. Phospholipids are essential for cells because they are major constituents of cell membrane. Their structure provides a classic example of how form fits function at the molecular level. A phospholipid is similar to a fat molecule, but it has only two fatty acids attached to glycerol rather than three. The third hydroxyl group of glycerol is joined to a phosphate group. Okay, so the two carbon of the glycerol, this is a glycerol molecule. Okay, the glycerol has three carbon skeleton. Two of them attach with ester linkage with two fatty acids. One of them has kink due to cis double bond between the two carbon. Okay. The third carbon of the glycerol attached to phosphate group, okay, which has a negative charge in the cell, okay, and then attached to an additional charge group or polar molecule is also attached or linked to phosphate group. We call it choline. The hydrocarbons tails are hydrophobic. The tails, those are the tails. This is space filling model. Uh, the black represent the carbon, and gray, those are the hydrogen, and those the oxygen. Okay. The hydrocarbons tails are hydrophobic. Okay, and are excluded from water. The phosphate group at its attachment form hydrophobic head. Okay, the phosphate group, because they are charged with negative charge, they form with choline, they form the hydrophilic head that has an affinity for water. When phospholipids are added to water, they self-assemble into a double bond, layer or sheets, as you see here. Those are the phospholipids molecule. They self-assembled in this way when we added them in the water. So we call this bilayer that shield their hydrophobic fatty acids, those, away from water. Okay. Uh, at the surface of a cell, phospholipids are arranged in a similar bilayer. The hydrophilic heads of the molecule are on the outside, as you see here, of the bilayer in contact with the aqueous solution inside and outside of the cell. The hydrophobic tails, so each phospholipid has two parts. head and tails. The head is hydrophilic and tails are hydrophobic. So the hydrophobic tails point toward the interior of the bilayer, away from the water. The phospholipid bilayer forms a boundary between the cell and its external environment and establishes separate compartments within eukaryotic cells. The third types of fats we have is steroids. Steroids are lipids characterized by a carbon skeleton consisting of four uh, fused rings. Different steroids are distinguished by their particular chemical groups attached to this ensemble of rings. Cholesterol, a type of steroid, is a crucial molecule in animals. It is a common component of animal cell membrane and is also the precursor from which other steroids, such as the vertebrate sex hormones, are synthesized. In vertebrates, uh, cholesterol is synthesized in the liver and is also obtained from diet. So, a high level of cholesterol in the blood may contribute to atherosclerosis.